types of solutions, and Pepti Systems has this knowledge. So in 2007, Lonza, one of the largest peptide manufacturers in the world, they requested for a similar instrument to be developed, but for peptide synthesis. GE started the project, put together the team, prototypes were developed, but then the project was shut down because GE needed to refocus on their core business, which was chromatography and not peptide synthesis. So the team then, what did they do? Well, they decided to start Pepti Systems. So far, the founders have invested about $150,000 in the company. We won the first prize in Shenzhen Entrepreneurship Competition earlier this year, and we got the Vinnova grant for Innovativa Startups Phase 1, as well as an ALMI loan just recently. The prototypes are ready, and proof of concept has been achieved with customer testing and verification of the technology to start now. This is our team. We have together over 100 years of combined experience in the industry, both in academia and working professionally in corporations. As you can see, many of us have PhDs. We have experts in flow-through column technology instruments. We have experts in developing software for these applications. We have peptide chemistry experts. We have market expertise because we've been in the industry for a long time. And also, I've been working in startups for a very long time, also within healthcare. This is our business model. It's quite easy, straightforward. We sell synthesis instruments directly to customers. We also provide consultancy and after-sales support, as well as offering complementary product sales to customers. We're using a proven product concept that comes from GE Healthcare that's proven on the market. First, a customer starts by buying a Pepti pilot instrument, which is a smaller scale instrument. They use this instrument to develop the optimal synthesis methods for producing a particular peptide. Once this has been done, they can order a process scale instrument and increase the scale to large scale production. The pricing has been determined by the product margin and the product cost, and we'll be manufacturing the process scale instruments with partners and the pilot instrument in-house. We'll also have payment in milestones on the process systems, which is industry standard, which helps us with managing our cash flow. The total peptide instrument market today is $1 billion, with both large and small scale instruments. The total addressable market is large scale instruments, and that's about $500 million. Now the total yearly target market for us is the amount of new synthesis instrument sales, as well as the amount of yearly replacements, and that's about $100 million. We estimate to take about 10% of the market by 2022, which is about $10 million. Europe and US are the beachhead markets, and China is the third target market. So what do we want in China? Well, we were looking for partnerships, customers, and of course investment. We're working with complementary product suppliers, one which is actually based in Suzhou, which is Ribo Biopharmaceuticals. We're allowed to sell their products as complementary in the international markets, helping them to reach new customers, and they can also help us sell in China. We're also looking to manufacture in China in the future. And we're looking for more complementary product partnerships in China. The ecosystem is large and growing for peptide synthesis, and there are a lot of manufacturers of peptides in China. We've done an online market study of over 500 customers. We've determined that the main potential customer for us is contract manufacturing organizations. They specialize in producing peptides and selling them to pharmaceutical organizations. They can have up to 30 batch reactors per customer, which means that just one customer for Pepti Systems can mean a lot of sustainable business for us. Biotech pharma companies are secondary targets because they're a bit more conservative, whereas contract manufacturing organizations are a little bit more open. So it is a conservative industry, and therefore you need to have a disruptive technology, and we have that for peptide synthesis. We also have a very credible team, which is very needed, much needed in this case. We're going to be working with contract manufacturers from the start. We're working with Polypeptide, Corden Pharma, and several others. They have confirmed our value propositions, and next they will validate our technology. The main competitors are using batch reactor technology, CS Bio, AAPP Tech, and protein technologies, and they have limited experience in flow-through column technology. So even if they manage to develop something similar to us, it'll take them many years, and by that time we'll already have captured a large part of the market. Recombinant technology is also another competing technology, but it has limited application because it can only be used to produce natural peptides, whereas most peptides actually have a synthetic component to them, so it's limited. We need about $250K to sustain the company until cash flow positive in 2020, in the first half of 2020. We're going to start customer testing soon. In June, final product development is expected to be finished, and first sales will be in Q2 2019. Then we will launch and sell. We'll also be looking to do more financing as we scale up our manufacturing after this. Thank you.
Thank you very much, Marcus.
Thank you.
You are keeping very good time. Thank you. I suppose now is the time for our judge. Do you have any questions?
Everything was crystal clear, huh?
So before the session, I was told if nobody have questions, then I have the responsibility to raise one question at least.
Okay. Please do. Go ahead.
We realized that you have a patent, no patent issue with GE Healthcare, right?
Yes.
This is clear with IP issues.
This is clear.
Yeah.
Yes, we already have a very good relationship with them, and they're aware of what we're doing, and they're very positive for it. They're a potential exit partner in the future.
Yeah. The second thing is, as I am checking a little bit on the Chinese suppliers in this same segment, also a lot of machine suppliers producing the synthesizing machine for polypeptide.
Yeah.
And for instance, they have sort of a base on the solid phase and the liquid phase synthesized process. Sure. So could you repeat again, what is your major selling point compared with existing technologies?
So first you have liquid phase synthesis, which is for smaller peptides, and then you have larger peptides between 6 and about 50 amino acids, which is done through solid phase synthesis. This is where our target is, so we're doing solid phase synthesis. Now, existing systems, they're using batch reactors. That's the main, like 95% is being done with batch reactors today. And you have large reactors where all the ingredients are put in. You have a chemist that needs to manually check each step. He needs to take samples, go analyze it, which takes time. It's manual draining of the, you have to drain the large reactors, which takes a lot of time, and it's not efficient. So you need to drain a lot of times, and you need to continue to wash this, which takes a lot of solvent consumption, which is very expensive. It takes a lot of time. The chemist's time is also very valuable. This causes a lot of carbon dioxide emissions. What we're doing is automating this process. So you're doing synthesis we're pumping the ingredients instead. So the whole thing is automatic. So you can just push a button, and then in a couple of days, the peptide is finished. Whereas otherwise, it takes months. And you have a chemist going and working day and night and weekends and so on. So we're basically pumping the amino acids through a column, and there the synthesis takes place, and then we can have automatic analysis of the synthesis and just see so that it's sure that the peptide is being synthesized properly. So cost savings, better product quality, and so on. So it's really revolutionizing for peptide synthesis.
Thank you. I may have missed it, but did you say anything about the length of the polypeptides you can synthesize?
No, I didn't. It's between about 7 to 50 amino acids in length is what we use for.
And could you compare that with the other method of synthesizing peptides?
Yes, we've done that already, and we've synthesized with better results than current technology. So,
is there a difference in length for the peptides?
Sorry?
A difference in length for the number of amino acids you can.
Well, we compared it with another peptide of equal. So we did synthesize a peptide with our system, and then one with a traditional system. And then we checked the purity and yield. And we were actually higher. And when we scaled up, we achieved even higher purity and yield. So we can, yeah. Does that answer your question?
Not quite. I was interested in whether there is a difference in length of the polypeptide chains you can synthesize using your approach versus traditional.
No, there's no difference in length. It would be the same length, yes.
Thank you. I think we're done now.
Okay.
Thank you very much.
Thank you very much.
我们第二位的项目演讲者叫Imran Abbas，他的演讲课题是 DNA 甲基化泌尿道肿瘤的早期发现试剂盒的研发。
Hi everybody, my name is Imran, and I will be talking about our project where we want to use DNA methylation for early detection of urinary tract tumors using urine as a sample, and our target population is Chinese patients. So, first, I would like to talk something about the incidence of urinary tract tumors. So if you see, here we have urinary tracts developed in bladder, prostate, and kidneys. And the prevalence together is almost 30%. Overall survival rate is very low. Why? Because majority of these cases are diagnosed when they are already at an advanced stage with low survival rate and less prognosis. And but these cases are curable if diagnosed earlier. This is exactly what we want to do, and it is important to develop an early diagnostic kit with high sensitivity and specificity, making use of urine as a test sample without any incision or something, biopsy. What is the current status of diagnosis right now? The cases, UTT cases, are diagnosed mostly with imaging systems, PET-CT or CT, MR, when tumor size is already 3 millimeters around, and it is at the middle stages with chances of metastasis when cancer has already spread. And there are no tumor biomarkers right now, no early detection kit available, and PSA is being used, but it is less specific early biomarker for prostate cancer only. Pathological examination, so it is mostly from middle to last stage when a pathologist can diagnose it. What is the project objective? So we want to introduce a biomarker-based kit for early diagnosis of urinary tract tumors for Chinese population. So if we look at the background, the milestones that were achieved are for this project are precise medicine and using epigenetics. Because if we look at the genomics, there are around 32,000 genes, and 8 to 10 mutations are required for cancer development. But in epigenetics, this replaces, and without any changes or mutations, only DNA methylation can cause aberrant expression of genes and results the carcinogenesis. So we can say that cancer is an epigenetic disease. So here, why epigenetics is important? Because if you see, this is a scheme of cancer development. So from normal tissue to cancer development and progression of cancer and metastasis, the epigenetic changes or DNA methylation occurs at an earlier stage. So if we can make use of changes in DNA methylation, we can target or diagnose cancer at an early stage. So here if we see that 0 means the patient, when the patient goes to doctor and is diagnosed. So if we had samples, for example, in this study, they used lung cancer so before 34 months, one can see the cancer development already there. So it means that we can diagnose lung cancer 15 to 35 months before its clinical onset using epigenetic markers. So what we have done right now in one of the related projects, we have developed a kit for early diagnosis of nasopharyngeal carcinoma. And you see that we have normal cases here and we have tumor cases here. So we can distinguish between tumor and normal cases. And here we used blood samples instead of tumor biopsy, and we can see the same pattern is being observed like in serum samples, and we compared them with tumor samples. Another project. So for this kit, for nasopharyngeal carcinoma, which is very much prevalent in China, so our detection rate was around 87% with 91% specificity. So we could diagnose out of 100, 87 patients. In one of the other projects, which was targeted for lung cancer, we developed same kind of assay for early detection. Here we had 6 tumor suppressor genes, and we used their methylation for early diagnosis, and we could detect 87% of cases with 94% specificity. So now what we want to do is we want to expand this experience, and we also had a pilot project where we already used urinary tract tumors and using our technique to diagnose those cases. But these cases were Swedish or European urinary tract tumors, and we could detect from normal to tumor cases. We could differentiate between these tumors. Now for this project, what we want to do is we want to establish the unique gene methylation spectrum of Chinese urinary tract tumors using urine samples. To re-establish and introduce a new early diagnostic kit which is specially made and tailor-made for Chinese urinary tract tumors. We also want to establish special magnetic bead method for extraction of traces of tumor DNA from urine samples, and we want to establish DNA methylation amplification method with new markers suitable for Chinese population. This is something about our team. So you can see we have decades of experience and knowledge about DNA methylation. We are based in Karolinska Institute, and we also have collaborators in Shenzhen, China, 2 different groups, and we have expertise in methylation studies and urinary tract tumors and genomic companies. So what we want to do, we want to establish a bank, 350 tumor sets. We want to perform DNA methylation and expression studies. We want to screen 300 confirmed UTT patients, and then we want to develop an assay and then use it for 1,000 urinary tract tumor patients, and then standardize and develop an assay and for the approval by FDA. This is our timeline. So we have already started working on that, and now we want to produce a kit up till 2021. Thank you very much.
I know you and Professor who has other projects in this area, in the lung cancer detection and so on. Good luck with that. Do you have any questions? Yes.
Thank you very much for the presentation. I understand this project is part of the Peacock project from Shenzhen. Maybe you can tell us a little bit more about how Suzhou is fitting to this scope, your venture scope in the future.
I'm sorry, could you please?
So how Suzhou, in this case, is fitting to your, let's say, future scope of the venture?
Yeah, because we already have got a grant from Peacock, Shenzhen. So now we want to expand because this is the same kind of an assay, but we will screen the same Chinese population, and we want to have some funding for this, expand and re-establish our already developed assay for this population. So maybe we can get some collaborators or
When would your test be used in the diagnostic pathway for UTT? And we're talking basically about 3 different types of cancers. Could you say a few words about that? Would it be used before MRI or PET scan, or when would it be used?
it could be used before for high-risk populations, or we can use it for before the clinical onset of the cancer. We would like to.
So you're suggesting it would be used as a screening
Yes.
for the normal population?
Yes, yes. For early diagnosis before cancer is there.
Before any symptoms?
Yes, yes.
Okay, thank you.
Well, it's my duty again. So you talk about these urinary cancers, and which include bladder cancer, the kidney cancer, and so on. So how when you do this pre-screening, how you differentiate? I mean, this could be a problem with kidney or.
No, it should